So welcome to session 9 of our mass spectrometry training series. My name is Rod Chalk, and today we're going to be talking about targeted phosphoprotein analysis. So we will look at the importance of protein phosphorylation. We will look at some of the limitations in phosphoprotein analysis. We'll look at phosphoprotein nomenclature, and we'll look at phosphoproteomics versus the targeted phosphorylation protein mapping approach. We'll look at a combined top-down and bottom-up phosphorylation mapping strategy. We'll look at using electron transfer dissociation. We'll look at some ways of measuring conformational changes as a result of phosphorylation. We'll look at enrichment strategies for phosphopeptides, how to extend coverage for phosphorylation mapping, and we'll also look at some real examples. So to begin, let's just remind you of the importance of protein phosphorylation. So what happens in protein phosphorylation is it's essentially addition of one or more negatively charged hydrophilic side chains onto an existing protein, and the function of that is to induce conformational change. So proteins can be phosphorylated, they can be dephosphorylated. Both of these are rapid and very efficient within the cell, and it's facilitated by hundreds of kinases and phosphatases, many of which have been characterized in the human kinome. So the way to look at phosphorylation is to view it, as I do, as a reversible molecular switch. So what it is doing, it's either activating or deactivating a protein at the post-translational level. So it's a much faster way of controlling things than transcription. It's the most common post-translation modification. It's been estimated that up to 65% of human proteins are phosphorylated, and that there are something in the region of 250,000 phosphorylation sites that could be mapped within the proteome. And many of these proteins, these phosphoproteins, are potential drug targets. So protein phosphorylation plays a central role in signal transduction, in receptor kinases and cyclin-dependent kinases. It plays an important role in enzyme regulation and also in protein-protein interaction. So we need to know about phosphorylation. Before we go into that, I just want to quickly ask you a question. Here is an intact mass spectrum of phosphoprotein. We know it's a phosphoprotein because this corresponds to the expected unmodified mass. We have 2 mass shifts here of 80 daltons, which correspond to phosphorylation. So the question is, how many phosphorylation sites are there? Well, you might be tempted to think there must be 2, but actually you may well be wrong about that. If we map the potential sites onto this data, so this is no phosphorylations, this is isoform 1, this peak could be any of 3 different isoforms. So it could be this one, it could be this one, or it could be this one. Okay, and this one could be this doubly phosphorylated one here. So it's possible to actually have any number of phosphorylation sites. Very often when phosphorylation sites map together, the presence of phosphorylation in one position will preclude phosphorylation in the adjacent position. But these 2 species will have exactly the same mass, so they will only produce a single peak. So phosphorylation state, this phosphorylation state 2 here, this defines the minimum number of phosphorylation sites, but not the maximum. There can be any number of sites. So please bear that in mind. And I want to just define some aspects of phosphoproteins. Phosphorylation state, as I've tried to illustrate here, this is the number of phospho additions to the ground state, the unphosphorylated protein, and it defines the minimum but not the maximum number of sites. So your phosphorylation state can be 0, 1, 2, 3, and so on. Phosphorylation site, this is the location on the protein sequence corresponding to a serine, a threonine, or a tyrosine. These are the residues that can be phosphorylated, where evidence exists for phosphorylation to occur. However, a site can be occupied or it can be unoccupied. And the sites may also be ambiguous or definitive, and I'll explain what those mean in a minute. So phosphorylation site occupancy, this is the extent to which a particular site is phosphorylated. And you express this as a percentage of the whole sample. Be aware that most phosphorylation sites are going to be less than 100% occupied. A phosphoisoform, this is a structurally unique phosphoprotein. It has a defined number of phosphorylations at a defined number of sites, and all of these sites are 100% occupied. So if you have isoforms with the same phosphorylation state, they will have the same mass. A phosphopeptide, this is a peptide which is known to be phosphorylated. So the phosphorylation state is usually known, and a phosphorylation site may be ambiguous or it may be definitive. So an ambiguous site is where the phosphorylation maps to a peptide which has more than one potential phosphorylation site, so more than one serine, threonine, or tyrosine. But where there is insufficient experimental evidence to specify the precise location. And then finally, a definitive site, this is where the site has been mapped to a single serine, threonine, or tyrosine. So here is the phosphorylation mapping challenge, and it is a challenge. So your phosphoprotein, it may be one of perhaps 10,000 cellular proteins, and it may be in low abundance. The typical MSMS coverage you would get on any particular protein would be somewhere between 10% and 50%. And the mapping that we do relies on MSMS. So PTM mapping, this is coverage dependent. So the chances of mapping any particular site is also going to be only 10 to 50%. On top of that, phosphorylation sites are usually less than 100% occupied. As I've said, they may be very low occupancy. They may be 1% or even 0.1%, and they may still be biologically relevant. And also, the vast majority of peptides in the digest that you will make, these will be non-phosphorylated. So there is a risk that these peptides will suppress any phosphopeptide signal you're looking for. So because of that, it's pretty much essential that you enrich. You either enrich for phosphoproteins or you enrich for phosphopeptides or both, and this is nearly always required. Another complication is the phosphate group is labile in collision-induced dissociation. So this can lead to ambiguity. So we know there is a phosphopeptide, but we can't tell which serine, threonine, or tyrosine it is associated with because there's more than one in the peptide. So this is ambiguity. Also bear in mind that you may be tempted to think that you can use negative ion because we're looking at negatively charged species. However, phosphopeptides, they may be negatively charged in solution, but in the gas phase, they always acquire at least one proton. And negative ion phosphopeptide analysis is not done, it does not work. I've just taken this out of Wikipedia because it's quite useful what it says. Basically, it says down here, bioinformatics analysis of low throughput phosphorylation data. So this is targeted data together with high throughput phosphoproteomics data, based mostly on MSMS, estimate that current high throughput protocols, after several repetitions, are capable of capturing 70 to 90% of total phosphoproteins, but only 40 to 60% of total phosphorylation sites. And you can begin to appreciate why that is. So here at the CMD, we use a targeted phosphomapping approach. And to compare that with more traditional approach, I've just compare and contrast these two techniques. So in a proteomics approach, we're looking at a tissue or a cell extract. In the targeted approach, we're just looking at a single purified protein. So in phosphoproteomics, the task is post-translation modification discovery. So we're looking to map as many phosphorylation sites in as many proteins as possible, or we're trying to quantify differences between samples. In the targeted approach, the idea is to map sites, but to do this to known phosphorylation states derived from top-down MS and kinase assay data. So we know that the protein is phosphorylated. We've got an idea of how many sites it is based on the phosphorylation state, and the idea is to map all of those sites. So in a proteomics context, site occupancy is unknown. In the targeted context, the site occupancy is known. So we know how many sites we're looking for. For proteomics, you are forced to use an automated data approach because your data sets are too large. For a targeted approach, you need to use a manual data analysis approach, and I'll describe that shortly. In proteomics context, there will be false positives. These are accepted, they're acknowledged in the false positive discovery rate. The number of false negatives in a proteomics experiment is not going to be known, but if you map any phosphorylation sites, your analysis is successful, and I put that in inverted commas. In a targeted approach, false positives are not acceptable. Also, false negatives are not acceptable. And in a targeted approach, your analysis is incomplete until you have mapped a number of sites which corresponds to the known phosphorylation state. So if you have a phosphorylation state of 3, you need to have mapped at least 3 sites. Otherwise, your work is incomplete. Before we move on, just to say something about electron transfer dissociation. So ETD, I've described this before, but ETD is a different fragmentation mode. The advantage of ETD is it is random, which is good, and the MSMS coverage is more extensive. And critically for post-translation modifications, phosphorylation and other PTMs will remain intact. So it's a more gentle technique, and there is potential for fewer ambiguous assignments. If you compare the two techniques, ETD versus CID, you can see the difference. The coverage is more extensive with ETD, it's more even, and the Mayo score is higher. So that's good from the mapping point of view. The problem with ETD is in early ETZ systems like the one that we have, although the fragmentation data is better in theory, in practice, the ion intensity for ETD is much lower than it is for CID. So when we have compared the two on a phosphoprotein, on a peptide, what we have seen is that the intensity for ETD is 35 times lower than it is for CID. So put this into context. We've said that phosphopeptide abundance is going to be low anyway in a mapping experiment. So if you reduce the intensity by 35 times compared with CID, in practice, what that means is phosphopeptides are not detected by ETD, not with our systems. They're quite some of the earliest ETD systems, and I think Later ETD systems are better, but this is the reason why we do not use ETD here at the Centre for Medicines Discovery. So I said enrichment is pretty much always needed. So you can enrich phosphoproteins. You do this using a proprietary affinity column. You would do this prior to digestion. But for targeted analysis, you would not do this. You would already have a purified phosphoprotein. Phosphopeptide enrichment, we do this using metal oxide affinity, but there are other techniques. You can use IMAX, you can use a strong cation exchange post-digestion. But you're generally only going to use one of these techniques because usually the amount of sample available is limited. So here at CMD, we use metal oxide affinity, and we do this with a mixed bed of titanium dioxide and zirconium dioxide. The reason we use these two selective agents is because they have partially overlapping selectivity. There is no enrichment strategy that will be selective for all phosphopeptides. Every phosphopeptide is chemically different, and you will only trap a subset of these. So because of that, it's also necessary to analyze the whole unenriched digest. You must do this as well. And I want to illustrate that with an analysis here. So these are two phosphopeptide analyses on the same protein. On the left here, we have 65% coverage and a good Maus score of over 1,000. And I've listed the peptides that we identified. The Maus scores are in brackets. And you can see we picked up one phosphopeptide here, this one. If we compare that with the metal oxide affinity enriched digest, the percent coverage is lower, it's only 44%. The Maus score is lower as well. What you can see is that the first peptide that we saw in the whole digest, we missed that. Okay, so that peptide was not captured on the affinity column. However, this peptide was, with a good score, an excellent score. Most of the so what the metal oxide affinity does, it will trap some phosphopeptides, but not all. It will also trap negatively charged, just generally negatively charged peptides. So you can see that I've illustrated here. I've just ranked these peptides by the number of negative charges and the pi. So it's necessary to analyze the whole digest and the enriched digest. You cannot rely on automated algorithms to identify all of your phosphopeptides. We use Mascot. Mascot will screen for phosphopeptides and it will find hits. However, the more post-translation modifications you allow in your Mascot search, the greater the number of false positives are going to be. And as I said, we cannot afford to have false positive identifications. So what's necessary is once you've done your Mascot search, you must validate every single phosphopeptide. And that's what we do. And this is the validation process that we use here. And we begin by looking at the Maus score. Okay, is the Maus score high? Well, what you want to know is the peptide real? So Maus score of 35, it's on the high side, but it's not spectacular. What you need to do is you need to actually analyze the data. And so this is the ion table for this particular peptide, and we can identify 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 consecutive ions that match the sequence. This cannot be arrived at by chance. So we are confident, although the Maus score is not massively high, you can be totally confident that this is a real peptide. Second thing you want to look for is neutral loss. So neutral loss happens during CID with serine and with threonine, but not with tyrosine. And Mascot will tell you if the software has identified neutral loss, which it has. You'll notice here that it's not 80 Daltons, it's not loss of it's a phosphate, so it's not 80, it's 98. So yes, it has identified a neutral loss, so that implies that we have a phosphopeptide. If you have used metal oxide affinity or some other phosphopeptide enrichment strategy, and you have captured your peptide on that column, that is circumstantial evidence that it may well be phosphopeptide. Generally, the only peptides that are captured are negatively charged peptides and also phosphopeptides. So it's being captured on that column. That increases the likelihood that the peptide is real. Do we have site ambiguity? Okay, so what this is asking is when you do a spectrum walk, and I'll show you that in a second, does this fix the phosphorylation state to phosphorylation to a single site? Another factor is when all the Mayer scores for the peptides are ranked, and is the Mayer score that we have, is the difference between that peptide match and the next highest match in the database, is it greater than 10? If it is, that also increases the likelihood that it is a good match, that it's a valid peptide, it's a valid phosphopeptide. And finally, consecutive ions when we do the spectrum walk, do these ions include the flanking ions either side of the phosphorylation site? So I'll just explain what I mean here by a spectrum walk. So this is the ion table from Mascot, and these masses assume that phosphorylation has occurred here at threonine 17. So all these masses in the table are correct if there is phosphorylation here. So when we look at the first mass in the B ion series, this mass 528, because we have this match, this means that serine number 1 and threonine number 4 cannot be phosphorylated. As we work down through these, the presence of these ions also are only possible if serine and threonine are not phosphorylated. This ion here, this tells you that tyrosine 13 also cannot be phosphorylated, nor can serine 15. Ion 16 tells you the same thing. We don't have this ion, but we do have the one after it. Okay, we have this one, 1813, and this tells you that this threonine 17 must be phosphorylated. So this is a spectrum walk, and this is what you do to make sure that the Mascot algorithm has worked correctly and the assignment of phosphorylations are correct. Sometimes they are not. So this is our Centre for Medicines Discovery targeted phosphomapping strategy. So we begin by determining the phosphorylation state and the phosphorylation occupancy, and we do that by intact mass. The next thing that we do is we will perform an in silico digest, and the purpose of this is to identify the optimum protease. You can use trypsin, but there may be reasons that, and you will be able to see this, why trypsin may not necessarily cut where you need it to cut to give you the coverage where you need it. So you perform the digest, and then we will analyze the whole digest by LCM-SMS. And the function of this is, first of all, to confirm that our digest has been successful, And if there are any high occupancy sites, we may be able to identify them in the unenriched digest. So the purpose is to identify any phosphopeptides not retained by enrichment. Okay, so we haven't done the enrichment yet, but this is to capture peptides that we would miss in the enriched digest. Also, because we haven't done any enrichment, we can quantify the site occupancy at this stage. Once we've done the enrichment, quantitation then becomes no longer possible. So we will enrich using this titanium zirconium mixed bed and analyze that enriched digest by LCMS. We will then collate the data. So we have intact mass data, we have whole digest MSMS data, and we also have a phosphopeptide enriched MSMS data, and we will collate all of that together. And that whole cycle will take 3 days. If we have not managed to map all the sites we were expecting, we would then choose a different protease and go through this cycle again. So here's a real example. So this protein MTHFR, this is a metabolic enzyme, and phosphorylation mapping was attempted on this some time ago by this group. And what they did was they did a sequence alignment from different species, and they were able to deduce from this alignment that these serines and threonines were highly conserved in higher organisms, and they assumed that this would be a phosphorylation control region in the protein. So this is our initial analysis. This is intact mass analysis. This is what the protein looks like untreated, and you can see that, well, you can't see, but these masses do not correspond to the unmodified protein. We've also analyzed the protein after it's been phosphatase treated, so we've removed some, but not all of the phosphorylations, and now we get a match. So this mass here, 75326, this is the unmodified protein. So this is phosphorylation state 0, 1, 2, 3. That's phosphorylation state 8 and phosphorylation state 9. So from the intact mass, this gives us a lot of information. It tells us that there are 9 phosphorylation states. So there must be at least 9 phosphorylation sites. And it also tells us that 8 of these are at 100% occupied. So that's good from the analytical point of view. Good to know that. This is the sequence. In this box, this is where we're expecting the control region to be. This is the conserved control region. And in the blue, these are the basic residues. So these are trypsin and arginine. Sorry, these are lysine and arginine, which are the tryptic digest sites. And so we can see from this that we would expect to get this tryptic peptide, which might be useful. This one's a bit too small to be useful, but this one might be useful too. So we perform the digest, and we've done this, and this is digest using smart digest trypsin. We then analyze the whole digest, the unenriched digest by MSMS. This is what we've done here, no enrichment. And then we enrich with titanium zirconium oxide. This is no enrichment. This is with metal oxide affinity enrichment. And here we've done it with a commercial tip, titanium dioxide tip, just to compare it with our internal method. This will generate our data set, and what we've done here is taken all the data for all the phosphopeptides and in fact all the peptide data and mapped it onto the sequence. So the coverage is yeah, about 54%. It's okay, But you can see there are large areas of the protein that we've missed, and in particular the area that we were interested in, this control region, we haven't got any MSMS coverage at all. However, it's not all bad, because we have we've been able to map 2 sites here. They are partially occupied sites, so we've got partial occupancy of these sites S3394 and 3N451. So the total coverage is 54%. We have not managed to achieve coverage in the region of interest. However, the data that we've got in terms of intact mass and MSMS data, this is in general agreement with the paper I showed you. They also failed to get coverage in this region. They claimed that they had coverage of 88%. And because they didn't manage to cover this region and they found no phosphorylation sites, their conclusion was that the phosphorylation must be here because everywhere else had been covered. However, what they were not able to do was identify these 2 phosphorylations that we found. So that means their actual phosphorylation mapping strategy is inadequate. Okay, so we need to do more than this. So we need to go through the cycle again. So second time round, we chose different enzymes. So the initial enzyme we used Smart Digest trypsin. Second time round, we used a combination. So we used chymotrypsin, we used pepsin. We can do that. In a proteomics context, you can't really use a non-specific enzyme because the database search would use up far too much memory. But we are just searching against a single protein in our database, so we can use non-specific digestion. And here we've used a mixture of pepsin and trypsin in our digestion. And so these are the modes we did it in. These are all in positive mode. This is the percentage coverage of MSMS we got. And these are the sequences in this initial region that we were able to map with some of the phosphorylation sites that were mapped. So this is the full data set. So this is the control region here, which I showed you. And here in red, this is the coverage, this alignment of all the peptides that we found with the phosphorylations mapped in them. So if we collate all of that data together, we get a composite MSMS coverage map. So this is with all the data from our using different enzymes and using either whole digest or enriched digest. And there's quite a lot of data here, and this allows us to get this composite MSMS coverage map, and we achieved 92% coverage for the protein, which is pretty good. And the encouraging thing is we were able to map through this region, which we could not map before. And you can see that there are phosphorylations exactly where you would expect them to be. Another thing that we can do is with MTHFR is we can use native mass spec to look at the charge envelope for the protein, and we can see how that changes in the presence of phosphorylation. So we can take an untreated phosphorylated protein, which is just shown here in the blue, and then we can do the same experiment after dephosphorylation. And what happens is that dephosphorylation, this results in a more compact structure. So this has moved here. The charge rate has got smaller, but also it's given us a more dephosphorylation gives us a more open and more flexible conformation. So in a second example, we're going to look at this cascade. So this is the BMPR regulatory cascade, which controls bone morphogenesis. And it's made up of 3 proteins which interact with each other, and they are all kinases, and they phosphorylate each other. So the first one in the series, this is ACVR1. This is a conserved region. This is the putative phosphorylation site that we expect to get. And this is the data that we're able to produce. So this is the intact mass data. And we can see phosphorylation state 2, 3, 4, 5, 6, and 7. So we're expecting at least 7 phosphorylation sites. And this is how we map the phosphorylation onto the sequence. And you can see here, this is the conserved control region, and we've been able to map 6 phosphorylation sites onto this region. 3 of them are definitive, so they're measured, they're labeled in blue, and 3 of them are, sorry, 2, I can barely see. 2 of them are ambiguous sites, and they are labeled in orange. If we look at the second component in the cascade, this is BMPR. Here is the BMPR intact mass spectrum. This is phosphorylation state 0, 1, 2, and 3. And in BMPR, we were able to map 5 phosphorylation sites here, 2, 3, 4, 5, with pretty good coverage. Okay, so that's our targeted phosphorylation approach, where we use in-depth analysis on a single protein, or in this case 3 proteins together. But what happens if we use an automated approach? Well, we know the answer to that question because we sent these samples off to a different lab where they used an automated approach, and this is what we got back. Okay, so this is the ACVR1. This is the control region, and here is the MSMS data. And we have coverage across all of the region. We have MSMS coverage over the region that we were interested in, the control region. However, there are no phosphorylations mapped in this control region. This is the second component, BMPR2, and what happens here is slightly alarming, because what we appear to have is peptides, flanking peptides here and here. Unfortunately, we know from the intact mass, and we also know from having sequenced the construct, that the construct begins here. So that we know its mass because we measured it, and the mass corresponds to this fragment, and there is nothing, there's nothing at a higher mass, so that we know that these flanking sequences, and also this and apparently N-terminal peptide, this cannot be present. We appear to have 112 post-translation modifications. However, if again you look at the intact mass data, what this intact mass data is saying is this is the unmodified protein, this is phosphorylation state 1, 2, and 3, and the other peaks here, these are sodium adducts. And what that means is that the only modifications that are present on BMPR2 are phosphorylation. These other modifications are artifacts. And just to illustrate that, here's the first one. This is formylation. This would be a mass shift of +29. We don't observe that. It only happens in the presence of methanol. This protein has not been exposed to methanol. And also formylation is not a normal modification for E. coli expression. This one, glyclamide, the mass shift is not observed. It's a chemical derivative. If you don't use this chemistry, you are not going to get this particular modification. And so this is false. This mass shift is not observed. This is also a chemical derivative. The same is true selenomethionine. You must have selenium present. There is none. This is an artifact. Biotin, this is also an artifact, and it just goes on and on. I won't bore you with any more of them, but just to summarize, It wasn't a complete waste of time to do this because we did manage to map 4 phosphorylation sites. These are similar to the sites we've already mapped. However, 2 of the sites that we confirmed in BMPR, they were missed. Okay, so so a third of the sites they were missed using this method. In addition, there are modifications which are in fact sample processing artifacts. So these are a result of the reduction alkylation procedure. There are also a further 93 incorrect PTM assignments. So what does this mean? If you have false positive post-translation modification assignments, what does it actually mean? Well, depends on what you're doing. If you are doing phosphoproteomics in this context, false positives, they are acknowledged in the false discovery rate. False discovery rate of 1% is acceptable. False discovery rate for PTMs of 94% is certainly not acceptable. False negatives, the frequency of false negatives, you will not know that. In a structural biology context, if you have a false positive phosphorylation or a false negative, this is not acceptable. This means your structure is wrong. So you cannot afford to be wrong in structural biology. So that's all I want to say. I'm just going to summarize. Phosphorylation, as I'm sure you're aware, phosphorylation is key in molecular signaling and it's extremely common. There are global approaches to phosphorylation mapping. These have been very useful, but they have severe limitations when applied to structural biology. This is because the algorithms that are used are unreliable and you are forced to use manual validation. This is absolutely essential. We looked at the reasons why people use ETD and reasons why we don't. We've looked at how phosphorylation can generate measurable conformational changes in a phosphoprotein. We've looked at enrichment strategies. We looked at extending the phosphorylation coverage, and we've looked at some real examples. So that's all I want to say. Just to remind you, next week we will be having the second of our guest lecturers, Dr. Will Greenland from Enrichment Technologies, and he will be talking about glycoprotein analysis. Please don't miss that. That's next week on the 26th of January, and I'll be very happy to take your questions.